So this is the second project, which is explainable uh, deep DT representations. So in this project, we'll be containerizing and creating endpoints for this project, explainable deep target representation for binding affinity prediction. So the problem statement in this paper was that given there are a lot of AI-based methods which are being used as machine learning scoring functions. A lot of them act as black boxes, and it's really hard for us to infer how exactly the decision is being made by an ML model. So in this paper, authors tried to uh, explain the reasons behind why CNN and followed by a fully connected network-based architecture model makes predictions. So authors use something called algogram. So they kind of create a pipeline where they analyze the weights and gradients which are being changed in the model architecture, and which will give them an outline of how essentially your model is prioritizing specific parts of your protein sequence input basically. So the input for this model will be a protein sequence and a smile string, and we also give it something like some sort of vector of values which indicates where the binding sites are, and some sort of vector of values where we are telling the model where PSSM scores are high and stuff like that. So for example, the sample input representation will look something like this. So we pass feature importance threshold, binding sites, window length, smile string, and protein sequence. So for this project, we created endpoints for evaluation and this algogram testing, because those two are the most the endpoints which will most likely be using whoever is going to use this API anyways. So for this, we create a post request. So the post request takes an item ID. So depending on whether this item ID is an eval or infer, we'll get two different outputs. So let's say if I make a post request to an endpoint eval, and the input is as such, we pass on name, description, smile string, the sequence information, the window, thresholds, and sites. And once we pass this and once we press execute, we get the evaluations basically. So we get an MSE, RMSE concordance index of the current trained model which is present inside the Docker image. So along with this, we also get a correlation plot, but right now we are not showing it, but we can see that it has already been created in the Docker image. And by using that correlation plot itself, these values are obtained anyways. So apart from this, if we make a post request to infer, uh, we will get the inference of this model. So basically, we get the weights where your the model thinks which uh, like when at what parts in this binding site is it prioritizing, like, and it basically we get something like a heat map again, that the map is present in the Docker image, but the y coordinates and x coordinates are we are this will be sent by the server as a response. So these are those values. So we can see that at when window size is 10, matching percent is 8, and when window size is 37, matching percent is 33. So we get so this list of values can be decoded back into a plot, and that plot is already there in the Docker image present anyways. So yeah, so for this study, we because the major takeaway is the inference of the model, as in we'd want to see where our model thinks are important sites in a protein sequence, we added that endpoint as well. So yeah.